Hey guys, it's Tiffany Martin with Medical Microbiology, and we are in Chapter 7, Microbial Genetics, and we are talking about DNA structure, kind of part 2. We're going to continue on from the previous video. Let's look at this double helix a little more magnified so we can see what's going on here. All right, so there is our double helix, and we have our sugar phosphate backbone. Remember that alternating sugar and phosphates, they kind of refer to that as a sugar phosphate backbone, and then our nitrogenous base pairs across. And so when we talked about pairing, do remember that this is just a shorthand diagram of actual chemical molecules. So there's carbons and nitrogens and oxygens and hydrogens, and they make this up. So if you go back to chemistry, you've got these molecules, and they're made up of these different elements, and they form the specific three-dimensional shape. So when we talk about A's and T's pairing up, it's because of the chemistry. How the different molecules, the hydrogens are put forth, adenosine and thymine are able to make two hydrogen bonds together, and that's how they're able to pair. Guanine and cytosine, they're shaped a little different, and when they look out, they have the space and the ability to form three hydrogen bonds. So this is why guanine can't pair with thymine or adenine. It just is not shaped correctly. And that's back to its chemical structure. So do remember that all of these shorthand diagrams that we're doing is based on a actual chemical 3D molecule and how it fits together with other molecules. Now my sugar phosphate backbone, this is made up of that deoxyribose sugar, because it's DNA, and that phosphate group, and these form covalent bonds. Remember, they share electrons, and a covalent bond is very strong. Comparison, hydrogen bonds, kind of weak. Hydrogen bonds are very weak interactions, and they're not near as strong as covalent bonds. Now, we said that this double helix is a very stable structure, and that's true. This covalent backbone makes it very stable. And one hydrogen bond, yes, is very weak and can be easily broken. But when you start linking up all of these hydrogen bonds in a row, it ends up being a very strong structure. And you can compare that to a toothpick. I can take one toothpick, one hydrogen bond, I can snap that sucker really easy. But if I put a whole handful of toothpicks together, and I've got like 10 in my hands, and I try to break all 10 of them together, much more difficult. So these hydrogen bonds individually are very weak, but when they form this double helix, it ends up being a very strong and stable structure, which is what you want from your instruction manual. All right, so as for the test, what I need you to know, I do not need you to memorize this kind of stuff right here. Again, if you know you've got biochemistry in your future, organic chemistry in the future, you may want to pay a lot more attention to it. But for purposes of this course and the vast majority of you, just know that this is based on the chemical structure, but we're talking about the shorthand. Now, this is one shorthand version here, this double helix ladder. But then what I do think you should be responsible for in this class is this type of diagram. So how the sugar phosphate backbone is put together, how it is attached to a nitrogenous base. And so that is what we're talking about with this sugar phosphate backbone. So this is sugar is has 6 carbons in it, and these, I think I'm going to switch to my diagram here. In fact, I'm going to start over from scratch so that you can see it drawn out. So it is a deoxyribose sugar, has 6 carbons and an oxygen, and when we draw it out, we have our oxygen, and then every time we get a bend, we get a carbon. So that's a shorthand version for a carbon. So there's 1, 2, 3, 4, 5 carbons. Oh, did I say 6? It's not a hexose, it's a pentose sugar. And there's 5 carbons. 1, 2, 3, 4, 5. Yes, 5 carbons in this molecule for that. And then that deoxyribose sugar